Hi, my name is Efrat Muller. I'm a postdoc at the University of Cambridge. And in the next 15 minutes or so, I'm going to try and provide a very high-level overview of multi-omic integration methods for microbiome research. The slides will be based on a review paper we recently published together with Tal Bamberger and Elhanan Bernstein from Tel Aviv University. So I want to start by saying that any of you who have worked with multi-omic data knows that there is currently this surge in computational methods for analyzing multi-omic data sets. And this is in line with our understanding of how valuable these kind of data sets are for understanding complex biological systems like the human microbiome. What you see here is just a small snapshot of review papers talking about multi-omic studies. So what I want to try to do in this video is to help provide a picture of the types of methods out there, and hopefully you'll be able to navigate it a bit more easily. So let's start with a few definitions and put multi-omic studies in the context of other approaches. So I'm going to talk about the gut microbiome ecosystem, but this is very relevant to any other environment. So when we talk about the gut microbiome ecosystem, we're talking about a very complex system with hundreds or thousands of different organisms, and they interact with their environment. They are metabolically active. They interact with one another. They interact with host cells, and so on. And we can view this very complex system as having multiple layers of information, each capturing a slightly different aspect of the system. Now, omic technologies, they allow us to profile some of these layers. So, for example, metagenomics allows us to obtain taxonomic profiles or functional profiles of our samples. Transcriptomics and metatranscriptomics allow us to profile expression of the different species or the host. We have metabolomic technologies and metaproteomic technologies that allow us to understand which small molecules are present in a sample and at what levels, or which proteins are available, and so on. Now, conventionally, to study a specific system, researchers take a very hypothesis-driven approach. So they look at a specific mechanism that involves specific features across these different layers. For example, a specific bacterial species that is synthesizing a specific metabolite that then influences the host in some way. There's also the single omic analysis approach, where we try to characterize a specific layer of information more comprehensively. So, for example, understanding the entire composition of species in a sample and how that differs between groups, for example. And then we have the integrative multi-omic approach, where we're trying in a way to combine both of these top approaches. So here we are trying to look at the system in a very broad and comprehensive way, but also look at connections between these different layers of information. Now, these kind of data sets come with a lot of challenges. First, there are the challenges that are simply because each omic by itself is challenging to analyze. Many of these omics are very high dimensional, with a number of features, sometimes in the thousands or the tens of the thousands, far exceeding the number of samples we usually have. These data sets are often very sparse, lots of zeros in our tables. They are often very sensitive to batch effects. There is the compositionality constraint, and so on. There are other challenges that are related to the combination of omics. So when we try to combine omics of different data types, that could cause problems for some methods. There are biases sometimes towards larger omics, so omics that have more features, and so on. One challenge, which I alluded to, was the challenge of deciding what computational approach to take in order to integrate the different data sets. There are dozens of methods to choose from, and it's also sometimes not very trivial to adapt a certain method to a specific study setting, study design. And lastly, there are challenges related to how we interpret the results that we get and how do we make sure that they are generalizable. So what I want to suggest in the next few slides is to categorize integration methods or approaches by the main research question that they try to target. I'll divide these possible research questions into four main categories. So first, we may be interested in understanding the relationships between the different omics. So putting aside any phenotype or other factor that might also be interesting. We can, for example, ask which omics are globally most associated to one another, which omics are less associated. We may ask what are the associations between specific features from different omics, for example, specific species and specific metabolites. And we may ask also if we can predict one omic based on another omic. All of these relate to relationships between the different omics. Um, I'll show a few specific examples for microbiome studies. So, for example, this is a study of healthy adults from a few years ago, and they obtained profiles of the microbiome from the gut and from saliva. They also had metabolome profiles from both gut and plasma, and they also had short-term and long-term diet profiles. And what they saw using a distance correlation test is that the gut microbiome is more influenced, more associated with a long-term diet profile. Um, Compared to the gut metabolome, which was significantly more associated with the short-term profile. There are other statistical approaches that can be used, for example, Mantel tests, which are very popular, canonical correlation analysis, and so on. Often we also are interested in one-to-one associations. So, for example, in this paper, they looked at correlation heat maps of stool genera versus metabolites and lipids. And there are a bunch of different correlation or statistical approaches we can use. Pearson or Spearman correlation. We can also use mixed effect models to control for various confounders we have, and so on. We can also look at these correlations through a network. So this is a multi-omic correlation network from HMP2 study. Different shapes of nodes indicate different omic features. And in this study, they analyzed this network in a bunch of different ways. Among other things, they identified hub nodes, so specific nodes that seem to be very central in the network. Another thing that people sometimes do is try to predict one omic based on another. Um, People can use machine learning methods to do this. There are also specific methods for predicting metabolome profiles based on microbiome profiles. And this could help us impute data, missing data for samples that did not have all the omics, for example, but it could also just tell us something about which metabolites are most strongly associated with microbial composition. Moving on to the next category, we may be interested in understanding how different omic profiles change given a certain phenotype or a disease. We may ask this at the global level, so which omic is most associated with a phenotype. We may want to understand whether a combination of omics can help us better classify samples or improve phenotype prediction. We can also try to identify multi-omic biomarkers of a disease that might outperform single-omic biomarkers. Again, a few examples from studies. So this is a very recent study of aging in a Chinese population, and they looked at different omics, different kinds of profiles, and tried to quantify how much each profile changes with aging. In another example from 2019, this is a pre-diabetic cohort, and the authors looked at whether or not combinations of different omics can help classify patients by whether they are suffering from respiratory viral infection. You can see here that by combining different omics, they were able to improve performance. This could be done using different approaches with machine learning. And there are other methods that try to identify these multi-omic biomarkers. So this is an example of an output from a method called Diablo. There are also machine learning approaches to do this, a very popular method called MOFA, and so on. The third category is more focused on the samples themselves and how do we stratify them? How do we possibly find clusters within the data? One very popular approach is called similarity network fusion. where we look at each omic independently and create this similarity network of patients based on that specific omic. And then similarity network fusion, SNF, iteratively unifies these different networks until it gets one consensus similarity network. And we can use this consensus similarity network to find structure in the data. For example, subgroups of patients that have a similar manifestation of disease, for example. This was done in several microbiome studies. For example, this is a study about patients living with HIV and using SNF based on lipidomics, metabolomics, and metagenomic profiles. They were able to identify clusters of samples that have similar multi-omic profiles and are also at higher risk of various metabolic complications. Lastly, we may want to focus more on possible mechanisms and use prior knowledge that we have in order to understand how different omics interact with one another. And there are different types of prior knowledge that we can use to tackle this approach. So one type of prior knowledge is metabolic networks. So this could be very useful when we try to integrate metagenomics and metabolomics, or metagenomics and metaproteomics. And there are a few methods that use metabolic networks to do this smart kind of integration. Another type of prior knowledge that we could use is biological groupings. For example, metabolic pathways. Metabolic pathways could be used to integrate, again, metagenomics and metabolomics, for example, by projecting both of these omics to the same kind of grouping space, for example, specific metabolic pathways. We could also use chemical classes or other kinds of groupings for this approach. Lastly, we can use our knowledge about the transfer of information from DNA sequences to RNA sequences to amino acid sequences. So there are a bunch of methods that use this information we have about transfer of information between sequences. For example, to integrate metagenomics with metatranscriptomics or metagenomics with metaproteomics. So this was a very quick and brief overview, but the take-home messages are, first of all, that multi-omic data provides a very comprehensive view of a biological system, and it could help us gain a lot of insight that we cannot gain through single omic analysis. Data integration is very challenging. We have all the problems of single omics and then a bunch of other additional problems. I would argue that the research question is the best starting point for selecting an integration strategy. So first try to articulate the question that you have and then try to find the best method for that. I would also want to suggest to try more than one approach and wherever possible, try to use prior methods that use prior knowledge as part of the analysis. Thank you for listening and good luck with your omic investigations.